This is part 2 of Chapter 12, Organization and Packaging of the DNA. In part 2, we're going to talk about eukaryotic chromosomes and chromatin organization. A eukaryotic chromosome is one long strand of double-stranded DNA from tip to tip. The short arm is usually referred to as the P arm, and this is the Q arm of a chromosome. And remember, there's one long strand of DNA that's wound around from tip to tip without a single break. Each eukaryotic chromosome contains many origins of replication, unlike the prokaryotic chromosome that we saw before. These origins of replication are spaced at approximately 100,000 base pairs along the length of a chromosome. So each chromosome has multiple origins of replication. In the middle of the chromosome, we have a structure called the kinetochore. The kinetochore is a collection of proteins that's bound at the centromere, which is the region of the DNA or the chromatin to which these proteins are bound. These, of course, are required for proper segregation of chromosomes during mitosis and meiosis. At the tips of the chromosomes, we have telomeres, here and here. And the telomeres are a very important sequence of DNA and associated proteins that are bound to these sequences that prevent chromosome shortening during replication. And we'll talk more about that when we talk about DNA replication. Eukaryotic chromosome compaction is based on the nucleosome structure. A nucleosome is composed of double-stranded DNA wrapped around an octamer of histone proteins, just like thread gets wrapped around a wooden spool. Here is the basic structure of a nucleosome. It has 8 different polypeptides, 2 each of H2A, H2B, H3, and H4. And they are arranged in a very specific pattern. And the DNA is wrapped around these core nucleosome proteins. In between each core nucleosome is a region of DNA called the linker region. A single nucleosome contains the 8 histone proteins plus approximately 146 or 147 base pairs of DNA. It is 11 nanometers in diameter. Okay, the DNA wrapped around the core nucleosome proteins produces a fiber of 11 nanometers in diameter. The linker region can vary greatly between 20 and 100 base pairs, depending on species and cell type within a given organism. By wrapping the DNA around these nucleosome cores, the structure of the DNA is decreased about 7-fold. Here's a ribbon diagram of a nucleosome structure where the DNA is in green and orange, wrapped around the core of the proteins, which are in these various colors for each of the 4 different types of nucleosome polypeptide: H2A, H2B, H3, and H4. How was the structure of the nucleosome determined? This is an interesting story, and understanding the experiment by which we were able to determine that DNA was actually wrapped around the core of nucleosomes is a very interesting exercise in understanding how to manipulate DNA and proteins. The model of the nucleosome structure was proposed by Roger Kornberg. He based his proposal on various observations about chromatin. biochemical experiments, X-ray diffraction studies, electron microscopy images, and a very important experiment by Marcus Knowles. Okay, this is Marcus Knowles' experiment. It looks complicated, but it's really actually very, very simple. Marcus Knowles started with a preparation of nuclei. Okay, so he took some cells and he isolated just the nuclei from them. And he put the nuclei into test tubes and he treated those nuclei with different concentrations of DNase. Okay, DNase 1 is an enzyme that will break the phosphodiester backbone of DNA and cut it into smaller pieces. Okay, so in the nucleus of the cell, the DNA is in chromatin. It is wrapped around the nucleosome particles. Okay, so before digestion, you have this beads on a string type structure inside the nucleus. When you treat with different concentrations of DNase 1, the DNase is able to cut in the regions that are located in between the nucleosomes. The nucleosomes protect the DNA, but the regions in between can be cut by the enzymes. So we let this proceed for the same length of time, a short length of time, in the 3 different concentrations of DNase 1. After the digestion, the DNA is cut in the linker regions. He treated these nuclei preparations plus DNase with detergent and phenol and extracted the nucleic acids, just the DNA itself, and ran them on different lanes of a agarose gel. The gel acts as a sieve. The larger molecules stay up high and the smaller molecules go towards the bottom. And you can stain the gel with a dye that picks up the DNA that's located in the gel. Okay? And you can view it and take a picture of it. So it's important to remember that in the gel, all the molecules of a specific length will coalesce at one spot on the gel. If they're small, they'll be towards the bottom, and if they're big, they'll be at the top, towards the top of the gel. So this is the data that he got. Okay, here's a diagram of what he interpreted the chromatin structure to be afterwards. The octagons are supposed to represent the nucleosomes, and the straight line is the region of DNA that's in between the nucleosomes. Okay, let's look at the high concentration of DNase first. At the high concentration of DNase, the enzyme will cut the DNA in between the nucleosomes and leave a fragment of approximately 200 base pairs. Here. And at high concentration, we can expect that the DNase will cut the linker region at every single spot that it possibly can, reducing the DNA and the chromatin to fragments of approximately the same length, about 200 base pairs. So at the high concentrations, DNase 1, all the chromosomal DNA is digested into fragments that are 200 base pairs in length. Okay, what happens at low concentrations? Okay, at low concentrations, you would expect that the DNase 1 would not cut at every single spot that was available because there wasn't enough DNase, not enough enzyme to cut at every single spot. So in our diagram above, it might cut here, and it might also cut one more time, say here, and nowhere else in this stretch of chromatin. So when we extracted the DNA and ran it out on the gel, we should find fragments that are 200 base pairs long, equivalent to 1 nucleosome's worth of DNA, but also DNA that's equivalent to 2 nucleosomes worth of DNA, 400 base pairs, and even nucleosomes that are longer, that are equivalent to 3 nucleosomes worth of DNA, 600, etc., all the way up until we reach the limits of a resolution. So here's our diagram now of multiple nucleosomes on a stretch of DNA. One thing to remember about nucleic acids in the cell is that DNA, RNA, is never naked. It always has some proteins associated with it. So in between these linker regions, there's two other types of proteins that we can find. Another type of histone called histone H1, and then some other large class of other proteins here called non-histone proteins. Okay, what does this H1 do to our 10 or 11 nanometer fiber? These proteins play roles in organization and compaction of the chromosome. They also play roles in gene expression. The H1 has a very critical role. If you look at DNA fiber without H1, it is our 10 nanometer fiber. It is just the beads on a string. DNA and a nucleosome, DNA and a nucleosome, a very long, linear, stretched out conformation. If you add the histone H1, all of a sudden you see a different structure. You see a further winding of the 10 nanometer fiber that we saw up here. So here's an actual electron micrograph of this now 30 nanometer fiber, this coiled coil, if you will, of chromatin that's found inside the cell. There are actually two possible models of a 30 nanometer fiber. This one is very tightly compact. It's called a solenoid model. And this one is called the zigzag model. Now, the only thing I don't like about these two depictions is that they don't actually indicate that in order to get the zigzag model at 30 nanometers or the solenoid model at 30 nanometers, you have to have the histone H1 here. Okay, so just remember, you need to have histone H1 in order to get this 30 nanometer fiber, either the solenoid or the zigzag model. Okay, that's it for now. We're going to talk about part 3 in our next video.